before lunch is going to be given by Tyler Bookinger from Michigan State. He's going to talk about genetic threats and opportunities as they relate to sea lamprey. So welcome, Tyler, as you move to the podium.
All right. Thank you, and I guess this is probably a little bit less of threats and opportunities, and more what I'll be doing is talking about some of the work that our lab is doing on developing the technical capabilities of genetic control for sea lamprey. And just kind of in this high-level overview, I'll be talking about a little bit of background, as well as some of our current goals and objectives and some examples of the approaches that we're taking, as well as highlighting some challenges. But I guess before I get started, I just want to say that I'm speaking today on behalf of myself and Wei-Ming Li, who's in the audience and will be joining for the panel discussion a little bit later. But also there's a larger team in our lab at MSU that is really important for this work, and then, of course, funding from the commission. So I wanted to start by showing this definition of genetic control, and I actually don't want to emphasize the definition as much as point out how encompassing this topic is, and kind of point out that genetic control is the idea of genetic control, and some genetic control options have been around for a while, including things such as sterile male release that would technically fall under this. But what's really fueled some of the recent discussions and opened up a lot of opportunities has been the development of modern gene editing technologies. And in particular, CRISPR-Cas9 is something that we talk a lot about and are using in our work. And just as a reminder, so CRISPR-Cas9 is a tool that's quite adaptable across species for editing the genome, where CRISPR allows you to identify a specific sequence in the genome that you're interested in, and that will then guide Cas9 or other Cas proteins to that area of the genome and cut it, after which you can either insert a different sequence or try to disrupt that gene function, which is a lot of what we're doing right now. And the fact that this is so adaptable across species kind of led to a lot of excitement and discussions in many different fields, but in this case, in how it could be applied for pest control. And a lot of this is kind of coming, and they're much further ahead of us, from like the mosquito and some other invertebrate pests, as is usually the case. So since this is so broad, I'm not going to touch on a lot of very specific applications. Although just to give a little bit of concrete examples, a lot of times we are focused on genes involved in reproductive processes such as sex determination or fertility. But I do want to just give a couple of general examples as we get started. And again, a lot of this is coming from work in mosquitoes. And what I want to do here is emphasize some of the spectrum in approaches that could be possible. And in particular, I want to emphasize the axis of how persistent these genetic engineering constructs could be across generations, and how likely they might be to spread. And on one end of this spectrum, we have the sterile male release, which has been around a while using generally not genetic engineering, but things such as chemical treatments. Although there is a lot of opportunity to use genetic engineering to improve this approach. And obviously, by definition here, this does not spread across generations at all. They're sterile, so they can suppress populations by competing with wild males for female reproductive effort, but it's not passed on. And somewhere kind of roughly in the middle, I have what this paper referred to as confined gene drives, where you can introduce individuals with a genetic construct, but for that to pass across generations, it has to be at a certain frequency in the population. And because of this, it limits its ability to spread outside where it's introduced, and also over time, if suppression wasn't effective, it would kind of naturally peter out on its own. And on the far other side, we have an unconfined gene drive, where in theory, even a single individual with this could pass that genetic construct on through biased inheritance, and it could spread rather quickly. And again, I just want to use these as general examples. There's certainly a lot in between here, and I know this is a little bit abstract, but I just wanted to emphasize the spectrum between kind of the one side of sterile males and things that are just kind of one-time things unless you maintain them, and then on the other side, unconfined drives. So with that, I want to switch over to C. lamprey, and some of the early investment that supports some of the stuff we're talking about now is the sequencing of the C. lamprey genome in the late 2000s, supported by the GLSC. But then, as was more generally the case in other species, the specific discussions around genetic control really were fueled by the development of CRISPR-Cas9 as a gene editing tool, and then shortly thereafter, the application of this in both C. lamprey and other lamprey species. So it was found to be an effective tool to do this in lamprey species. And this led to some initial assessments of whether genetic control was a direction that should be pursued. And this work was supported by the GLSC and led by Thresher, Jones, and Drake. And in a couple of papers, they assessed this question in a number of different ways. First of all, through engaging with stakeholders to see if there was support for R&D into genetic control. And these stakeholders included, probably many of you, but agencies in the Great Lakes who are either directly or indirectly involved in fisheries management, and then also the broader fishing community. And the second couple of things they did were to form expert panels and kind of assess the technical feasibility and perceived risk of different genetic control options, and then also do some modeling to evaluate whether these could potentially be effective for population control. And altogether, they found pretty widespread support, both from stakeholders and also scientifically, that at least some of these genetic control options were worth pursuing as far as research and development goes. So after that, the GLSC included research into potential genetic control in their strategic vision. And then also around the same time, there was a new research theme focused on genetic control, under which our lab and others are doing this research now. And in the strategic vision, they kind of laid out two outcomes, which I'll roughly paraphrase as seeking data to answer the question of can we do this, and then seeking data to answer the question of should we do it. And our lab is focused on the first question and developing the technical feasibility, though recognizing that there's really a lot of broader questions that need to be answered at that point. So getting into what we are specifically doing, our overall goal is really to leverage gene editing technology to, first of all, better understand C. lamprey biology, but then also develop the techniques to potentially leverage this in a control program. And again, emphasize that we're focused on the technical feasibility. Within that goal, we kind of have two general objectives. The first one, which is really a major focus of ours, is starting off to identify the genes that are associated with traits that could be exploited in control. And this is important foundational work, because we don't, in C. lamprey, being that they're not a model species for this type of work, we really need to start by just identifying which genes are important. And in particular, we're focused on several reproductive processes, including sex determination, what makes larvae become male or female, and then fertility. And then kind of a second objective generally is focused on some of the more the tools and techniques that we would need. And here, like, gene editing is a well-developed tool in other species and has been adapted successfully in lamprey. But in the context of control, there's some questions that and issues that would need to be overcome before it could be useful. So I'll highlight one example for each of these in a couple slides. but I wanted to start by just giving a general idea of what this work is looking like for in our lab at least right now. And we do almost all of this at MSU. Certainly all the gene editing is done at MSU in a lab that the GLSC supported the construction of. And this facility has tanks set up for bringing the broodstock in and raising embryos, and then raising them for several years through larval development, together with some of the gene editing equipment and supplies that we need to study gene function in those larval development stages. And all of this is set up as a recirculating system within MSU, and then goes through a decontamination unit in our basement, and then enters the sanitary sewer system. As far as the CRISPR-Cas9 experiments, what these look like, we kind of start off through a number of approaches like litter searches and transcriptomics to identify genes we might be interested in. And once we have gene sequences that we're interested in, we can work through commercial vendors to buy a guide that will bring our Cas9 scissors to that location in the genome. And so we can just buy those reagents, and then we inject those reagents into embryos when they're at the 1 or 2 cell stage, so right away, pretty much after they're fertilized. And these are injected like under a dissecting microscope. My hands are way too shaky to do it, but some of those other people on our team lead a lot of that work. And then if our gene edit is successful, this would then disrupt the protein that would be the product of that sequence, and then turn into differences in the phenotype of those individuals. So just to give you a little more concrete example of what we're talking about here, this is a gene that we just use as a model to develop some of the basic tools. Tyrosinase, it's involved in pigmentation in fish. And here you can see I have on the left zebrafish, which we use as a much quicker model for a lot of our work. And you can see that knocking out this gene, you get albino zebrafish, and then on the same thing here on the right where we've done that with sea lamprey. So this is a phenotype that's pretty easy to observe, but not one that is necessarily relevant to control. It's just a useful tool for developing the techniques. Also want to take a moment to highlight some of the challenges with this work, and the first and one of the major ones is the life cycle of sea lamprey. As you know, it's quite long. And an example of why this is challenging is, as I mentioned, a lot of our work is focused on sex determination in larvae, and in their long life cycle, they don't really even start to develop gonads for a couple of years. It's more related to their growth rate, so we're trying to accelerate that, and certainly benefit from the SLAP work that Fraser mentioned that John Human's involved in. And then the seasonal spawning is really difficult. Basically means we have that summer window to at least initiate all of our experiments, so we're getting to a pretty busy time here. And then just the complexity of their life cycle makes it difficult too. Again, the SLAP work is really exciting for us, but as of yet, it's still difficult, and I'm not sure anybody has raised them through like the parasitic stage in the lab, so it remains difficult to raise their whole life cycle in the lab. Another set of challenges is related to their genome, which I'll just refer to as messy. It's rather large and repetitive, and it actually changes over time, where in early development they ditch about a quarter of their genome. And these things have led to there being relatively few genomic resources, which are really critical for these types of experiments. So a couple specific examples. First, for that first objective of identifying target genes, again, we're really interested in sex determination, and this is really a focus of ours because a lot of the different genetic control options all require data on the genes that are involved in this. So it's not really directed at one one specific potential application, but many of them. And at this point, we don't know what factors, genetic or environmental even, influence sex ratios and sex determination in lamprey. So some of the things that we're doing to try to answer this question include transcriptomics, where we've isolated cells from a developing gonad and are looking at gene expression to see if we can identify genes involved in male or female development. And then based on that and literature search, target specific candidates to knock out and then raise them to see what the effect was on their gonad development. And then, like I said, their gonads don't develop for a couple of years, so we're also working to develop some early indicators to see what the effects of these experiments were on gonad development. And just a quick example for the second objective, developing some tools that would need to be developed for control. One thing we're focused on is the scale of editing, where, like I mentioned, we currently have to microinject each embryo to make edits, but there are other options that could potentially be more scalable, and one example is electroporation, and this has been used in zebrafish and other species where you kind of have a little well of many embryos, and you introduce an electrical current and it basically pokes holes in the membrane and allows those reagents to go in. We're working on this in sea lamprey, but as many things, they present their own challenges, and their chorion is rather thick and a little bit difficult to penetrate, so we're working on that right now. So just to wrap up here, I think the first step towards this work was the initial assessment, engaging of stakeholder support, and considering the risks and the potential benefits of this. And after that, I think we're doing the next work, kind of developing some of the technical capabilities that would be needed here. And I think that's kind of one of the more sea lamprey specific steps. So looking forward a little bit, I think once we answer some of the questions we're focused on, I'd be optimistic that some of the next steps might be easier to adapt from other species, especially mosquitoes, which here I have a picture of in Brazil. You can just buy a little box of genetically engineered mosquitoes, which obviously that's not where we are in this country, but certainly there are species we can look to learn things about the process. And lastly, again, highlight the importance of that second outcome in the GLSC's strategic vision of evaluating some of the past the technical side, some of the questions that will need to be answered. And with that, just acknowledge funding and support and the broader team that's working on this, and I'll take any questions.